No, good morning, still morning. Yeah, it is a great pleasure for me to welcome Dr. George Stuart to offer us a distinguished lecture in the Center of the Causal Discovery. George finished his bachelor degree in the science and BA and BS in the University of Colorado, Boulder, and he finished his PhD in biomedical informatics at the Stanford University. Josh, I guess I got to know Josh by reading many of his papers. Because currently he's the professor of he's a department of the biomolecular engineering, right? All right, yeah. I got to know Josh by reading many of his papers, and I got to know him at the conference. Josh mainly is working as the intersection of the computer science and cancer genomics and he's a leader of the major cancer genomic initiatives, particularly the well-known TCG projects. Right now, he's leading a group to do all the pan-cancer genomic analysis. So, published many of the seminal works. It really has been a pleasure to read, to learn all those things. So it's great to have him to come over here, give us an update on what is going on. I think he's going to talk about pan-cancer analysis and other things. Without further ado, thanks. Welcome.
Thank you very much. Pleasure to be here. It's a real pleasure to be here visiting the center. I mean, you guys have some of the luminaries and founders of causal graphical models, and I studied that stuff for my PhD work at Stanford, so it's just great to be able to meet them. I got to eat with Greg Cooper, who was like one of my heroes last night. So it's humbling and inspiring all at the same time. But yeah, I want to talk. I have a two-part story you'll see today for the talk, and some of it's going to mention the pan-cancer work, looking at lots of different tumors, and then I kind of want to bring it home to what do we do for individual patients to make it to make this kind of more discovery effort, we bring it home to the clinic, like why are we doing all this, and so forth. So first, as the title suggests, I mean, I wrote the abstract you saw a while back, but I'm going to take you to some newer stuff and launching from there. So a lot of talks these days start with this figure. This is like the most used figure from NCI, the precipitous drop in DNA sequencing, but it's the background of my work, a lot of our work. DNA sequencing is sort of just changing the picture every month, it seems like. But I used to show this to my students, right? So if you see, this is an old plot. It goes to 2014, and everybody was talking about the $1,000 genome. It's going to cost $1,000, and it looked like it plateaued there. The bottom of the line is $1,000. This is per genome cost, right? It started out with the first human genome project that cost $100 million, right? So it's so much cheaper now to sequence a genome. But then if you just download it a year later, the same graph from NCI in 2015, you can see that there were breakthroughs in technology again that actually made this a reality. Now, I like to say it kind of costs the same amount as your phone to sequence your genome, and this is what I talk to students about in our genomics class, right? Is there's this idea of the negative dollar genome. If you keep extrapolating this, and that actually is a real thing, right? So probably you'll start seeing companies pay you for your genome, right? Because it's just this useless list of letters, your genome. I mean, the sequence of it is, and what you're going to pay for is the apps that interpret it, right? So a lot of what we do is going to be on figuring out what is actually useful information in that DNA sequence. Was there a question already? You guys are already alert.
Yeah, can you tell the lawyers and the regulatory compliance people that it's useless stack of letters?
Yeah, we should. We actually need some of these algorithms probably to interpret all the legalese and those kind of documents too. My collaborator David Hausler likes to use this slide just to kind of drive home, like if you saw the same kind of drop in pricing in the auto industry, we could all buy a Ferrari for like $50. So I mean, it's been amazing, DNA sequencing technology. And I'm preaching to the converted here. We want to be able to use all this information we're going to get from the genome projects for medicine. And it'll be kind of a bittersweet story. Some cliffhangers today for you, like to be continued. I feel like and you'll see the way the clinical care is set up right now just can't respond yet to what we can do with the information. And that's sort of the sad point we're at. So we're a little ahead of medicine at this point, maybe for a good reason, but we can maybe have a discussion on that. So the simple idea, right, the thing that sets us off on this investigation is to think about what's different in the tumor than the normal. And so the basic idea is sequence the tumor, sequence the normal, see what the differences are. Sounds easy, right? Of course, there's errors in our sequencing technology still, so you do this over and over again, and the first challenge is to kind of figure out what's the noise and what's the signal, and that's even a hard problem. And I'll actually kind of hint at that because it was a tripping point, still is for a lot of the investigations we want to do. And you have your big, massive DNA sequencers that can churn out a lot of these reads. A lot of us work with read level data, or if you're like me, I'm a systems biologist. I'm way downstream of the processing, and I need somebody to tell me what genes are altered, right? So I can do my pathway analysis. But if you could sort of look at the databases and what they're holding now, and you could plot them. I mean, here's a little rendition of it. And I used to show this kind of to sell Santa Cruz as the purveyor of this information, because we used to run CG Hub. Many of you probably used it. It's now, last summer it kind of got decommissioned, and it's now run by University of Chicago. But the center sun there is sort of the size of the cancer genomics data relative to some of the other genomics data sets that a lot of us probably recognize, like the ENCODE project over there on the right. And then 1,000 Genomes kind of came close with 220 terabytes, but we're at like more than 5 petabytes now, right? This is an old slide. University of Chicago's got probably 12 petabytes or something like that. So the information coming out now is sort of, right, the picture you should have is we're getting more like every year than we had, in all previous years of information that's being deposited. So there's many informatics challenges there of their own kind, right? How you store it and retrieve it and all this kind of stuff. Here's a really old slide from way back when they first published the first human genome, right? They were like, okay, how do we assemble this thing? Or we sequence it, so how do we put it together? Same kind of thing today, right? We're sequencing all these cancer genomes, and it's like, what's important in them? And Xinghua and his group, of course, are really working on different aspects of this problem. So, some of the stuff that people work on, that my lab works on, TCJ is interested in, right, is, and I'm not even listening, even just detecting where the changes are is tricky. But once you do, which ones have functional importance, I'll mention a little bit of that. And you'll see a bias in my talk. I am heavily suggestive that we also have functional information. I know Xinghua would agree with me. The RNA sequencing information, it kind of gives you a readout of what pathways got hit in the cell, or as you'll see, the environment that the cell's in, the tissue, or the immune cells around it. So you get a lot of information out of that RNA. A lot of the clinical practice doesn't know this yet, so part of my beef these days is to get that conveyed to the doctors and the health caretakers. And then and I think what's relevant for your center, right, obviously, is pathway analysis, causal structures are very important to help us interpret these data. At this talk, I'm kind of showing you the sneak peek of where I want to get to. I'd like to have a little model for every tumor, and maybe even better, every little subclone in the tumor, right? As you guys all have heard about heterogeneity, and there's a family of tumors. But imagine you had this little wiring diagram for each tumor, and you could then kind of, and what this would mean would be, red would mean this is active in the tumor and it's not supposed to be, because it's active in the tumor and it's not active in normal. And if it's blue in that plot, it's inactive and it's supposed to be active, right? So you kind of want to reverse the logic in that wiring diagram somehow, and you'd be able to, pick your things that you could inhibit. Your kinase is what you have drugs for, and so on, to pick a way to reverse that picture you're seeing in the tumor. All right, so how do we get to that? I'm going to just tell you this little two-part story. Like I mentioned, we're going to try to, there's no statistical power in trying to build a model for one patient sample, okay, at this point. So we're going to try to use and leverage from the many that we have in the database. Part of the story is, what are the different types of cancer, and then identify that type that a patient has, and then refine on that theme after you've got the subtype. So that's sort of the bias of the approach I'll advocate today. So with the TCGA project, and there's also a related effort in the international community to collect a lot of data, to then analyze all the tumor specimens and kind of figure out their commonalities and differences. And so, I just kind of picture this fake mathematical space, right? You've got, the pathways that are altered, that's sort of the top axis, and then the cell that got hit, and there's different wiring within each cell, which genes they're expressing, makes them vulnerable to different types of mutations. And then you also have the mutagenic soup that the cells are sitting in, right? That they can get hit by different carcinogens and so forth. And from all the data that we have, we can, land or address a tumor in this space. And then the hope would be to take other data, right, like treatment outcome, is what I'm showing here, and then be able to just predict, are you going to respond or not to a drug, depending on where I put you in this mathematical space, right? Anyway, I usually have to show that to clinicians. I know you guys are mostly mathematically oriented here. All right, so as Jinghua mentioned, I was put in charge of this pan-cancer project a few years back for TCGA, and they collected lots of different data on all these different data platforms, and I'm just showing you the bird's eye heat map view, right, of that. Expression data is over in the upper left, mRNA expression. They also had microRNA expression. We had some protein level information. It's not proteome wide, but some key players and known players in cancer. And so that's the kind of data you have here. You go to a TCJ meeting, or you listen to any of the calls, and you hear about all the different analyses of all the different platforms, and then you hear other folks talking about how they use 2 or more platforms, right? They'll say, we looked for genes that were amplified and their expression changed, right? Or they were methylated and then their expression went down, or they're methylated and their copy number changed, or there's all these ways that you can look at it. And in fact, right, that grows combinatorially with the number of data platforms. And I'm a little more of a simpleton, so I kind of wanted to think, is the gene on or off? Is it active or not in the tumor? And when I would go to these talks or listen to them in the beginning, I would just feel like I was sitting at this light, right, which I just stole off the internet. I don't know where this intersection is, and hope I never run across it. You sort of get information overload. You don't know, what to make of the data. And kind of the analogy would be like if you didn't know anything about planetary motion, and you look up at the star pattern, the patterns of the motions of the stars, you'd see all these correlations, and there's all these different ways of connecting the data together. But then once you know like a simple rule, and it's not that simple, I realize, but like mass is the determinant of the motions. That's what's driving the gravitational forces. It helps you, simplify all those correlations that you're seeing into a simpler, interpretable picture. And so, this is the Center for Causal Reasoning. You guys will appreciate this, right? Before I joined TCGA, they'd already published this paper on glioblastoma, and they showed this diagram that Larry Donenhauer made. He's a P53 expert, but what it told me was, and how you read it is each of the circles is a gene, and they're kind of writing down how many patients have an alteration in that gene. And if you look at any one gene, right? Like, I don't know, can people see the mouse? I'm not sure. I'll just have to talk about it. Like in the upper left, you have like CDK4, 18% of the patients have an alteration, right? Or, you know, possibly a different subset, half of them have this deletion in CDKN2A and all that. And then some have mutations in retinoblastoma, 11%. If you look at any one patient, it's like they have a different problem, but if you know that all of those genes actually ultimately screw up cell cycle progression, then you've related all those, all those patients together. You now understand. You have a better picture of that they do have a common problem. So the pathway level helps you interpret and coalesce that, right? Very nicely. I mean, so now you know like almost 80% have some problem in retinoblastoma, right? Or almost 90% in something in P53. And the cool thing is one, I guess lots of genetics and biochemical research over 30 plus years has collected these relations about how genes interact with each other and how they depend on each other and so on. And then they've been databased by places like Reactome and like I'm listing here. So you're kind of standing on the shoulders of giants here. We don't have all pathways described, but there's still plenty to make sense of some data. So when I first got involved with TCGA, we wrote this method called Paradigm, which is a factor graph model that combines the different types of data you have and then allows genes to kind of influence each other. And I'll just talk for like 1 minute on this. I've given whole talks on Paradigm, but basically what you have is a model where the variables here represent some state of gene expression, right? You have a certain number of copies, certain amount of expression, a certain amount of protein, if the protein's active or not. Those are kind of hidden variables in this model. And then you have little constraints between them. That's what these factors are, these little black boxes, right? So, and those are just they specify, high probability and low probability configurations of the variables, right? So if you don't have any copies in the cell, then it's a low probability you can express anything from that gene if there's no copies for the gene. That's sort of what these factors do for you. And then you have data that you can hook into right? So your observed data is shown in gray here. You have microarray data or RNA-seq data now. You can hook it up, and the factors kind of tell you how to transform the reading on a chip to internal state of the mRNA kind of thing, right? And so in this way, you can sort of let all the data kind of influence you to tell you ultimately is the protein active or not, which is maybe what you care about. And so if you get some expression data, here's P53, and in your pathway it says it activates apoptosis or programmed cell death. And if you see that P53 is expressed, the belief propagation will say, well, then you're more likely to have some protein. If you have protein, you're more likely to have active protein. Now you're more likely than the prior to go into apoptosis. That's what this belief propagation would tell you. The nice thing is you can handle interactions between genes. So here's an inhibitor for P53. It actually promotes the degradation of P53 protein, MDM2. So you can hook up the active state of MDM2 to how much protein kind of gets made. So you can see it's connected to the factor influencing the protein level of P53. And so now if you see the same data for P53, you have this sort of counter logic that says, well, I know you said you've got mRNA, but you also have mRNA around for MDM2, the inhibitor. So now I don't know, maybe you're not likely to be in apoptosis because that protein is going to get degraded. So you can come up with a completely different answer based on the gene activity. And you can come up with a score based on sort of a log posterior odds kind of thing. And that's Paradigm. So we've run it, and I've given the network to biologists, and this is what it looks like, right? It's crazy and awful, and you don't want to like sit down by your fireside with it and curl up with it like a good book. It's pretty awful. It's a hairball, and my friend Larry Hunter calls it the reticulome, which I like. But computers can actually do some computing with it, obviously, and some processing. So we've contributed quite a bit to looking at different subtypes of cancer, and you can see what's like for example, like aggressive, non-aggressive, or so forth, and you can find are there like subnetworks in there that are implicated and so forth. And one of our first contributions was on the ovarian paper, which came after GBM, to show a little mitotic regulator involved in that, and sort of a crosstalk between proliferation and repair, and how maybe the wiring's messed up in ovarian cancer cells. And that just was a huge hot signal from the paradigm analysis. From all those different data sets, we created this integrated data, right? So I always tell my students, don't make data from data, because it's kind of going backwards, but that's sort of what Paradigm does. And what it is simplifying that we can now just use this one data matrix. And the data matrix is, again, samples by, and now we have pathway entities, and they're sort of inferred activities. They're made up by the computer, what's active and what isn't. And we do our machine learning on those things. And so one way we're viewing the data, which I think is a lot easier to see, and a lot of people have used dimensionality reduction techniques, and t-SNE is another way to view these things. We used a force-directed layout method here for the samples, and then we visualize it in a map that uses Google Maps technology, actually, that we're getting ready to publish. And what you see is colored here are the samples from that original pan-cancer study. So there were 5,000 patient samples, and you could look at the integrated pathway activities, and this is what you got. And we were kind of bummed out, actually, when we saw this, right? Because most of the tumors follow their tissue of origin. It was like, if you didn't have to give us any data, we would have kind of guessed that. And when we looked and counted, like 90% of the samples were with samples from their same tissue of origin. The flip side was 10% weren't, right? And so, well, hold on to that thought. So first we had to sort of kind of validate this map meant something. And so if you zoom into different tissue types, here's like breast cancers. You can see it separates out some of the major subtypes of breast cancer. Basals and luminals are there on the red, and the blue over there on the right. You can see some HER2 amplified tumors shown there as well. They're the yellow dots on the right-hand plot. And then the cool thing about this tool we have is we load it up with all these attributes, and then you can do just differential analysis of what's clustering together. You can just take advantage of spatial statistics, just abuse them, and then since it is a map and it's a layout, you can just ask, what kind of attributes have values that cluster in the map, kind of thing, or two attributes values that cluster together in the map. I mean, all this spatial statistics is already there. And so if you just run this little spatial statistics, you can see that estrogen receptor signaling is higher in the luminals compared to the basals, kind of thing. Just kind of pops out for these layouts. So it's fun to use it. And we've shown it for a lot of different tumor subtypes within each tumor kind of get recapitulated on this map. So it's kind of nice. I won't go into those just for issues of time. So back to this pan-cancer map, I said 10% were mapping to different areas than you expected from the tumor type. So the best example is these bladder cancers, which you can see I've restricted on the right-hand side. There's a bladder island set, and there's some that are mapping with head and neck squamous tumors, bottom left, and then some of the lung adenos. And then you can look at the patient outcomes for just those bladder cancers. You can see, again, the blue plot, those patients that were in the bladder island had better outcomes than the ones that looked more like squamous or lung adeno disease. And I guess the point with this is that the pathologists had already looked at all this data, and they couldn't tell for those bladder cancers that they looked squamous-like, right? So the RNA data and all that other data that went in there, that was a little more sensitive, right, than the microscope. So it's just sort of, if you could get that information into the clinic earlier, they could treat those patients differently than just run-of-the-mill bladder cancer. They're due to smoking, or from a squamous cell kind of alteration. So so that's kind of point number one, is that this molecular data could be important for treating the patients. Obviously, they had different outcomes, and you know, I'm very interested in leveraging that kind of data to get it to the patients as quick as we can. So the chapter 2 on this, and Ling-Hua mentioned this, that there's a pan-cancer analysis going on now. So TCGA ended in the summer, and so we're all on sort of afterburner now. It's like the last project is to analyze all the tumors all together, and now there are 33 different types instead of just 12 like I showed you before. And you can see there's about 1,100 samples in this data set. We've built some Google Maps with it. We're still analyzing it. But you can see there's a little more mixture going on, which will be kind of cool to think and reason and figure out why. I'll show you one example that, I mean, I could understand, and that's why I picked it out. So that, you know, I wanted to see a pan-cancer cluster before, and I didn't really see one. And this is what I would call a pan-cancer cluster, right? The one in the middle, the blob of cells that are from lots of different tissue types, right? But what you can also see is there's sort of these lines, right? It's kind of fed from these other tissues around it, right? And then there are lines of samples kind of running into that center cluster, right? I don't know if people want to guess what that is, but I've had some people guess it when I show this plot. But so there's it sort of says that there's a mixture going on, right? There's a mixture of your tumor type, and then there's this other thing shared among all these other tumor types that's a signature driving the similarity of those samples. And it turns out that it's the immune component. So if you ask this geospatial stats, what's the best thing? It finds like this is a T cell program is one of them, and then an interferon program for, you know, we have gene signatures that recognize these things. And you can see the blob down there. It's also present in other tumor types, but it's pretty highly activated in that center thing. So there's immune infiltrate in those tumors, and so you have a mixture of the cell of origin signal with the immune response. So that one made sense. So there are the bladder cancers now in the current map, so they're mapping to more than just like 3 areas like I showed you before. There's more like, I don't know, maybe 6 or 7, depending on how you count. So it'd be interesting to think about what those bladder subtypes are. I mean, you have more tumors now to compare them to. So so before we thought we were estimating 1 in 10 were mapping differently, and we think now it's going to be like 1 in 5 will have pan-cancer connections. And to me, that just means there's plenty of surprises in here and room for the data to tell us something than our expectations going in, and that could help a lot of people. So what we did to sort of make this a proactive tool rather than just, you know, a static library of tumor data was we started a pilot project where we would take a sample where we had RNA-seq data, and this is for pediatric cancer samples, and we would look it up in the map, and we would try to find, okay, what's the closest form for that sample, and it's part of this treehouse project that we have for pediatric cancer. And as our pilot study, we analyzed 7 samples, and these are from children who have failed, you know, very high doses of radiation and chemotherapy. Usually that works for kids, and they can tolerate that kind of treatment. But when it doesn't work, nowadays they'll get they'll do some exome sequencing, and they'll look at the variants, and they'll see if that tells them anything. But often it doesn't. You have these variants of unknown significance. And then they get the RNA data, and then they're like, okay, now what? Now what do we do? Does the RNA data give you some kind of clue about what you can do for this patient? And so we partnered with them to look up some of these samples. And so the white dots here are what we get when we look them up on this Google map, right? We just show this visually, but behind the scenes, of course, it's this n-dimensional space. And what we're doing is we're looking up the neighborhood of those samples in this map. And we have the TCGA data in the map, and we have some pediatric cancers, like from Target and some other collaborators that contribute to it. And Sometimes you don't see anything surprising. You can't help them out. Like the middle plot there with the squamous cell. There's a squamous cell carcinoma and a map to squamous, head and neck squamous tumors. That didn't really help at all. But there were some surprises in here. I'll just tell you about one of them. So there was a sarcoma, and that mapped to this neuroblastoma land. And that was kind of interesting. And when we look at the neighbors of those neuroblastomas, they had a particular fusion in common in this gene called ALK. It wasn't like the best thing. It was like, top. It's like the fifth from the top or something. And we're not allowed to suggest treatment, but we can just show them this is what characterizes tumors like the one that you gave us. And the doctors look in there and they say, oh, there's a ALK fusion. And then you can look at the ALK expression status in this patient. That's shown right there, TH001. I recoded it because I'm not allowed to show you the actual identifiers from this precision oncology trial even. But you can see their level of ALK was kind of on par with those that have a fusion in ALK, and much higher than other sarcomas, which is shown in the blue there. And that was actually enough for the doctor, and they were looking for, they were expecting that this 8-year-old boy only had a few weeks to live. So they gave this boy crizotinib, which is an ALK inhibitor, and he actually failed on that after a year, and then they gave him a JAK inhibitor, and then that worked for another 6 months or something. JAK's kind of in the same pathway, and it's usually a resistance pathway of ALK. So that was that was a good story and a bad story, right? I mean, we think that the boy's life was significantly extended, but a sad story because the whole system isn't responsive enough to get us more RNA data, right? We couldn't reanalyze the recurrent sample, and maybe we could have seen that there was something suggested in the next sample that we would get. I'm not sure why that we can't get that, but there are other issues at place there. It was enough though to be encouraging to us that that we expanded this effort and we were funded by this precision medicine initiative in California, and our part in that is called the CKCC. It's the California Kids Cancer Comparison, and basically it's set up to do just kind of what we described. We serve on a tumor board meeting every week. Actually, it's usually 2 or 3 patient samples, and we they give us the RNA-seq data. We look it up in an ever-growing map, and we have to prove that some robustness with kind of like bootstrapping, and we can tell them, yeah, the neighborhood is stable and so on. And when the neighborhood's stable, sometimes the neighborhood isn't stable, but when it is, we tell them what's overrepresented in the neighborhood of a particular kid. And you can see, I don't know if you've read this while I'm talking about it, but it's the same kind of thing I was saying. They'll have some exome sequencing, maybe it's from a targeted panel, and in addition to the RNA-seq data. And they're thinking about all these things and including their previous treatment history and so on. We looked it up in the map and showed them where it is, and then kind of give them a description of what's overrepresented in the neighborhood. And that's basically where we have to leave it, and so that they can kind of pick from there and weigh it with other evidence what to do. So it's a little, and then the next part of my talk that we're going to move to now is. And it's a little quicker. It's saying, we're treating each of these samples kind of generically, right? You're kind of looking up their tumor with this collection. You're the closest match. But every tumor may have the possibility of having its own nuances, its own set of mutations. It could have its own alterations. Maybe the reasons some people fail or don't ever respond is because they have some little uniqueness to their tumor. And so that's sort of chapter 2 here. What can we do to sort of fit a model better to each of these patients? All right. So just a reminder in the beginning, we want to get that pathway knowledge. And what we're going to try to do is connect the events we see in a patient with the RNA sequence data in a way that we get some kind of pathway model that describes what we think is going on in their tumor. And the first thing really is to kind of weed through and figure out what's a driver and a passenger. You don't want to do this you don't want to use all the variants that somebody tells you about a patient sample, because a lot of them are just there. You can pick up passenger mutations in sort of genes that you don't need in that tissue. And so I know Jinghua's group has done quite a bit of work on this, distinguishing between passengers and drivers and so on. This is a very active area of research. Some people call it the needle and the haystack problem. I think it's harder, so I call it the needle and the needle stack. It's one, identifying the variants is part of this, and then two, it's identifying which ones are functionally important. Even just identifying them, I mean, this is just a little aside, which ones are there versus errors. The TCJ project had very little overlap, even in just identifying somatic single nucleotide variants. That's the plot on the left, this Venn diagram. At that point, we had 3 different centers doing the calling, and you can see the intersection in the middle is about as big, maybe even a little smaller than the unique things that they were calling, right? So the callers, this is just detecting variants. We're coming up with more unique things than there were common things. That was alarming. And then for structural variants, it was even worse. And so some friends and I got together and said, we need to, we've heard about these dream competitions, so we launched one. I was sort of the right person to do it because I don't, everybody didn't think I was going to cheat. I don't develop methods to detect mutation variants, so I was sort of an impartial judge, I guess. And the shocking thing to me was the amount of participation we got in this was, and I just had no clue. When we closed the contest, this was a couple years ago, there were 345 contestants. And we've since left it online, and people kind of benchmark their methods, and it's over 500 now that are developing just variant detection algorithms. Like a lot of these dream contests, we could show like the wisdom of the crowd thing, where the upper plot in these, this is accuracy of calling, and you see the gray line is an ensemble made up of multiple methods. There was a clear, really good method. The dot all the way to the left is UTech from the Broad Institute. But you could still kind of improve on the Broad's method if you kind of combined, 4 or 5 callers and so on. So anyway, that was the dream competition. I guess one other aside there is that the thing I like about these competitions is you get to see negative results, which you hardly ever get to see in science. It's like everybody's submitting all their predictions to you, and they don't, and you're evaluating them, so you get to see all their screw-ups. And then you get to analyze the screw-ups, which was fun. So one of the screw-ups, this is false positives. There was a pattern. I don't know if you guys have heard of these mutation signatures, but a lot of the algorithms were slipping up and calling mutations where they shouldn't have. We knew where the mutations were because some of our data was synthetic. We'd injected the mutations, and a lot of callers were predicting mutations where we hadn't injected mutations. And you're like, well, why would that be? And we think what it turned out is that there's some library artifact, some deamination going on that's sometimes in the normal, sometimes in the tumor, which is what we based our simulation on. And if you have that with low read coverage, you can trip up some of these callers, and they tend to see this kind of deamination thing that you might think is an age-related signature that was kind of published by Stratton a little while ago. So once you know the variants, then picking out which ones are functionally important is another level of challenges. And there's a review that Rachel Karchin wrote, and I just wanted to show you guys that they're sort of at the point now where they're looking at consensus. Do we have consensus of these methods that try to gauge the functional impact of these variants? And so you can see that some of them do. On this plot is from their paper, just different algorithms for measuring the impact and how much they overlap with each other. And so they're sort of at this point where they're trying to build consensus on that. My lab did a little bit of this and took a pathway perspective on it. So if you think about a gene that's mutated, like FG is focus gene, if you run something like I talked about before, Paradigm, you kind of get a mixed picture, because if it's mutated, what happens is you'll see that the downstream effects of the gene are low, and because of feedback circuitry, the cell will try to rescue it. And so you'll see that like the regulators can be high. And so our pathway, this Paradigm method would sort of give you a wishy-washy picture of, you know, we knew P53 was knocked out in this patient. Well, why isn't Paradigm saying P53 is off? It would be somewhere in the middle. It was kind of embarrassing. We'd always show P53, and it's like, your method's broken. It should show that it's off. Well, a student, Sam Ming, came along and said, well, maybe that's because of this competing circuitry thing, and came up with a method that predicts whether there's a discrepancy in the signals downstream and upstream. So he runs this method I talked about, Paradigm, twice, where he takes a gene and he disconnects the parents off of it, runs it once, takes the gene, disconnects the targets off of it, runs it again. And those of you who know about like causal interventions in graphical models, you'll see that we're doing a hack, and we didn't do this correctly. And I'd love to talk about how to do it appropriately. But so we ran this twice. We have these mutilated graphs we run in, and if you get a different answer in these two different runs, we call that the shift score. So we can call it paradigm shift. Okay, so we have this paper that if it goes up, right, it means that you have higher activity than you expected from the regulators. You weren't supposed to be on, but you are. You're like oncogenic, right? You're doing something and you weren't supposed to be on. And if the shift is lower, it means that you know, your regulators are screaming high. They want to turn you on, but your targets aren't doing anything, and so you're loss of function, right? And it works for a lot of genes really well. So like for retinoblastoma, it's sort of a known tumor suppressor. The outer ring there kind of shows you the inference of this method. Blue is lower, so loss of function in those patient samples. Each tick in this plot is a different patient sample as you go around the wheel. I'm not going to spend too much time, but in the pathway models, look at all these double negation logic. I won't draw you through it, but you know, like the inhibitors of the inhibitors are predictive of upstream regulatory status kind of thing. And then there's another example, NFE2L2, known oncogene in lung squamous cancers and others. And you can see it's mildly red on the outer ring. And so what we could do is sort of plot this discrepancy or the shift score, that's the y-axis here, and rank all the mutations that way. And you can see NFE2L2s on that plot over there. It's not even the highest one, right? And in parentheses, I'm showing you how many patient samples. So the cool thing with this is it doesn't really depend on the sample size. You know, a lot of these methods that infer the impact of a sample kind of look at how often does it happen across your cohort, right? So this is a nice orthogonal way to look at the mutations, whether there's an imprint on the pathway around the mutated gene when you have the mutation. Okay, so now we have to believe ourselves that we have, for a patient, we're given a set of mutations. We can kind of weed away the passengers maybe, use multiple methods, not just paradigm shift, to find which ones may be functionally impactful. We have the gene expression data at the bottom. We want to kind of connect those things together to come up with you know, what we think is the logical diagram that explains the tumor data that we're seeing. And so you can use methods like Andrea Califano's got this marine algorithm, this master regulator analysis, and it can take the gene expression data, use what's known about transcription factors, and sort of link a transcription factor to the targets that it thinks regulated that gene expression signature, right? That's sort of what Marina does. So you can get one level up, right? You can get to the transcription factors. There's even a hierarchical way. Now you can ask, what kinases might explain the activity of the transcription factors, activity that you inferred, right? Which is another way to stitch these together. What we published a few years back now is this algorithm called tie-dye, which is in the same vein as Joel Bader's got some network diffusion methods, and Ben Raphael has HotNet. What this does is it puts different sets of genes on a network, a given network, and then lets them diffuse on the network, and it looks for where they're mixing. And the places where they're mixing are the places where the things are getting linked from one set to the other. Like, so one of the sets is the set of mutations you saw, and the other could be the active transcription factors. And then the purple things here are the kind of the signaling molecules maybe that help transfer that signal, right, from the disruptions in the genome to the disruptions in the transcriptome. Right. So we had a we had run this on some metastatic prostate cancer models, and on our Stand Up to Cancer Dream Team, we had a collaborator, Owen Whitty at UCLA, who had done like proteome-wide phosphoproteome and serine, threonine, and tyrosine kinase mass spec analysis of metastatic prostate cancer and primary prostate cancer. And so we could then ask, if we built one of these wiring diagrams for a generic metastatic prostate cancer, okay, does it tend to link in the things that Owen's lab thinks are active in metastatic prostate cancer, right? Because they're measuring directly, and we're trying to infer it. So we sort of had a way to test this hypothesis. And so what we would look up is, well, are our linkers inferred to be active on the phosphoproteome data? And it's not perfect. You can see this red plot is sort of the, how active those are as a distribution compared to the things we don't link in to the metastatic prostate cancer network. And there's a slight shift, right? Statistically significant if you run this KS test, but it said that maybe they're kind of looking at similar aspects of the data. You could also kind of do a marina analysis of Owen Whitty's mass spec data. You could say, well, you saw these things phosphorylated. What's supposed to phosphorylate that, and is it active? Like AKT is supposed to phosphorylate a bunch of other things. So we would infer the regulators of the phosphoproteome data. This just shows you an example of some of those. And then you could then look up, are the regulators more active in these networks that we're trying to link up as well. And I wouldn't be up here, but of course that worked out as well. There was a slight enrichment there. So it told us that maybe we could, if we want to build a metastatic prostate cancer network, we could use the phosphoproteomics data, we could use the genomics, the transcriptomics, run our network diffusion algorithm using some big background network, and build a generic network for metastatic prostate cancer. And so the cool thing with this is you're kind of using the network to integrate everything, right? So you have all these different studies that had different data sets for metastatic prostate cancer. You had some gene expression, you had this mass spec data, you had some genomics. The Venn diagram just shows you like there's zero overlap, meaning no sample had all of them, right? So we're just trying to get as much data as we can about metastatic prostate cancer. And we build, in this paper that we published this past summer, we call it the Scaffold Network, okay? So this network is supposed to draw from this big generic network and kind of distill across a lot of metastatic prostate cancer patients, these are the genes that get altered or phosphorylated, or, right, they're transcriptionally modulated and so on. So that was step one, and we used tie-dye to do that. And then the final step is, and I'm almost done here, so if you haven't paid attention to anything, pay attention for the next 2 minutes. So we're going to take the patient's own data, that's on the left. Then we're going to take as the input network that Scaffold, the generic network. So before we did this, right, we matched that patient to its closest subtype, and we have a network for that subtype. In this case, it's metastatic prostate cancer. Okay, it's a certain type. I'm not going into this type. It's kind of a neuroendocrine type of prostate cancer. But so you have that subtype, and then what we did is, and I think there's much more room for improvement on this, and I'd love to talk to you guys about it. We're just going to do it because this is the method we had. We're going to run diffusion again. The patient's data on now this metastatic specific network, and you get like the reticulome again, right? I'm not going to show you guys the huge nasty network, but this is for one patient. So patient 40, and I'm just showing you little like photographs of it as we kind of went around this network diagram. And I'll just walk you through one example, right? So patient 40's network. Take a look at the right-hand side. All right, so you have, and the way you read this is, you have APC, usually mutated in colorectal cancer, actually. This patient has a mutation in APC. Not only that, it has a deletion in the other arm. So they have like loss of heterozygosity in APC, which could be interesting. Hyperphosphorylation of beta-catenin, that's the CTNME1. And then you have a polychrome complex gene that's amplified. Is that, yeah, amplified, EZH2. Okay, so And then those are DNA methyltransferases, and they're also modulating. That's also a chromatin marker. What I haven't shown you here, which is kind of sneaky in the background, is AKT1 in the middle there, right? It's all grayed out. I need to make another slide so what we did, and we need an algorithm to do this. There's no formal algorithm yet. Well, we have a hokey one here. It just says, take out the hubs, right, of that patient's network. So you infer this network. What's interlinking all these alterations that you're seeing? Well, in the middle of it all is AKT. So put that to the top. And then, okay, given AKT is in the center, who's next in the center on the left side and on the right side kind of thing. So what we did is we make a tree diagram out of these patients. And it turns out that often the center of one patient's network was the center of another one as well. So the way you read this is AKT was at the top, so was SARC, that's on the left over there. But then after you considered AKT, then there's this other kinase, PRKDC, and so on. Now, how do you use this? And I'm going to end, this is the last example. So here's patient 40 again, and you can kind of plot out its activities on this tree now. And now you can say, well, if you have a molecule that inhibits AKT, then maybe you can treat with AKTs because it's going to shut down that whole right-hand side or whatever of the network. If you're a different patient and this is patient 30, and this poor person also has kind of not just the AKT branch but the SARC branch, that one you might suggest, okay, that person maybe needs two options here, or maybe the bottleneck PRKDC if you have a molecule for that. So you guys, that's how you read this tree out, is you want to kind of shut off all the activities by picking off who's the master regulator at the top and so on. So I didn't show you this, but we demonstrated this in cell lines first. Okay, we had to treat cell lines like they were patient samples, and we tried this, and then we knocked out the thing suggested at the top, and we had like 3 cell lines we could use. Okay, and I think like 2 out of 3 it worked or something like that. So the cliffhanger that I mentioned is we're just putting that out here for these deceased patients, and that this paper just says if you had this information, you could have maybe considered treatment this way, and it just leaves it there, right? So it's sort of unsatisfying that way, and it's just really just an algorithm, but it's pretty ad hoc as well, and there's a lot of interesting causal reasoning that should go on in here. For example, like you're thinking about an actual formal perturbation on a graphical model instead of me lifting up and doing a topological sort. It should be like what gene actually reverses this thing maximally, reverses all the inferred activities the best. I mean, so there's plenty of work to be done there. So in summary, this is kind of what I talked about, right? So you can take this data, map out the forms. I showed you that we've got this tissue-driven aspect. I was excited. I was talking to Jinghua. That he can look at more of a tissue-independent type of depiction of the cancer samples. There's a whole TCJ project I mentioned that wants to do this now, so we need algorithms for that to get at the more subtle signals going on from the microenvironment and the immune environment. But even still, you can see some interesting connections with those. Yeah, it's even the tissue does carry a lot of information anyway, and it's important, I think, for treatment, and it is helping some of these pediatric cancer patients now. I didn't show you any of this data, but we can show that proteomics data really did help us infer the metastatic prostate cancer networks in the cell lines where we could test it out. It was very, very helpful. So we need methods that can kind of pull in heterogeneous data from some samples and use them for others where we don't have that data. I think that's really a promising area. And we need these models that can kind of inspect these nasty networks we come out with to make things more interpretable. So I'm still interested in how to interpret which mutations are impactful, and I think there's plenty of work there to be done. And then I showed you a couple kind of hacked together ways to identify pathways for patients. We'd love to do it on deconvoluted data. I'm playing around now with some methods to formalize how we pick what genes to knock out, like these little, like I said, weaknesses here. But that that, I mean, for this group, that really is a causal type of analysis that needs to be done in an interventional model. And then everything is sort of moving toward more refined pictures of these tumors from at the single cell level. So I have a bunch of work there that I'm not talking about now, but it's a huge new direction for me. Building up signatures of pure cell states and then going and analyzing tumor specimens with those. So the folks in my group this is my direct group that did the work. Evan now moved to the Califano lab in Columbia, is the guy who wrote tie-dye and did all that patient-specific network analysis. And Yulia, who just graduated, made the Google Maps and helped us establish that collaboration for using it for pediatric cancer. And this is all done with my collaborator, David Hausler. We have a genomics institute at UC Santa Cruz, and we partner with the Buck Institute north of San Francisco. Chris Benz is our breast cancer expert who works with us on most of these projects. So thank you, and I'll take questions if there's time for it. Thanks.
Yeah.
We got a couple minutes, right?
Yeah, well, we have some time. Yeah, maybe.
Is everything crystal clear?
What is the basis for the network clustering? I still don't quite.
Which part?
So, for example, you cluster the different type of cancer by network clustering.
Right. Oh, when I showed you the Google map, like the samples and
what is the basis?
Yeah. So when we run Paradigm, I showed you that matrix. We get activities for all the things in the network, and that's just a vector for each sample. We just compare them with, pick your favorite metric like Spearman correlation, and then treat those. Our method treats those as spring constants, and you solve this energy minimization to, this force-directed layout I mentioned to just satisfy the springs. But you could use, like, we've run Tisney to plots, and you get pretty much the same answer. So play with different metrics, and you get pretty much, but that's because tissue is such a huge dominant signal that I think almost anything you do right now, until we figure out how to deconvolute, it's going to do something pretty similar.
These are based on what type of data, or like the combination of different data?
So the map I showed you was mRNA expression, copy number, mutations, and methylation, DNA methylation. Kind of get rolled into that model. But for the PanCan study, you can use and we show this in the paper, you can use almost any platform alone and you get very similar answers. And then we had 4 different integrated methods. One's iCluster, one we came up with called COCA, and then I forgot the name of it. Paradigm was one of them that I showed you, and then I forgot what MD Anderson called theirs, but they all got basically 95% similar answers. So, but again, it's because tissue is just such a driver. Yeah. Was there another question in the back? I thought I saw it.
But one of the things I think is really awesome about the Cancer Genome Atlas too is that you have a repository of normal tissue information. I was wondering if you've done any work trying to use the sequencing information and transcriptome information from normal tissues to identify the tissue-specific signal and then filter that out.
Yeah, we haven't yet, but that's clearly what we're going to do next on this working group is we have a good pool of normals for most of them. Some of them you don't, but and then you can borrow things like GTEx data maybe where you don't have it to get the RNA signatures for the normal and subtract it out totally. I've had like a couple rotation students do it, and just to share with you, the first thing we did was we did do that. We subtracted off the tissue using like a linear decomposition kind of thing, where you try to still maximize the variance in the data, but account for the variance across the tissue types. And then she reclustered and made a map. And bizarre to me, I saw more tissue. They're still clustering separated by tissue, and there's more subtypes. It's like now I have like 5 clusters of breast cancers, and 4 clusters of bladder cancers. And I was just like, I didn't expect that. I thought there'd be mixing.
Well, that
now,
almost makes more sense though, right?
Yeah.
Because maybe when you don't account for the tissue-specific effects, it's just masking all the subtypes.
It could be, and those subtypes are still distinct for each tissue, which is, but I just didn't believe it because it was like a rotation student. I was like, maybe you ran something wrong, but I don't know. Maybe it's the real deal. But I think I usually only believe if we do it like at least like 3 different ways. But anyway, it's a good idea.
Yeah, I think previously we do not fully appreciate the tissue-specific or particular cell type-specific mutation events and the pathway utilized. It's a little bit difficult to really get rid of them. And I just think that maybe you have more experience. Some genes, every cancer utilize the existing systems in the given cell type, right? Seldom a cancer invented some pathway that's never used in this particular cell to do things. Mm-hmm. And so I think that what you found is a similar cancer type. It's just really hard to distinguish.
Right.
Some part of the disease mechanism are also cancer type specific.
I think it's yeah. Well, I think that approaches like you were showing me before the talk are the way to go. So develop signatures of specific types of mechanisms that go off in different types of cells, different stages of development, or different mutation statuses and different tumor types. Build models of those gene signatures that are specific, and then you can go back and look at all this data where it's mixed both populations and see if you see that you see a clue that signature is in there, even though it might be multiple signals on top of each other. You might see a signal of that specific type of disorder that you wouldn't detect otherwise. I think that is a valuable way to do it, to go that hasn't been tried enough yet, right? Because then we'll be able to connect more tumors with grossly different tissue of origin signals with, you saw the same pathway alteration because you have a signature for MYC amplification or whatever it is. And there is a MYC amplification signal in these tumors, but the tissue signature was so strong you wouldn't have related them without doing it. I like your topic model that it's getting. That's a totally good way to go.
All right. Thanks.
Yeah. How do you like that? I just fed back your research to you from your question.
All right. So maybe we have more opportunity to collaborate.
Yeah. Anyway, all right. Well, thank you, everybody.
All right. Thanks.